Well, I think we moved to another area of autistic perspective. The next presentation will be on special tests for children with development difficulty. So, may I call upon Dr. Ming Xing Ma. At present, he is now the General Manager and Technical Director of Kunming Huanchi Biochip Industry Company Limited. Dr. Ming Xing Ma graduated with BSc in Biotechnology from Yunnan University, and later on he had receiving Ph.D. degree in Biochemistry and Molecular Biology from St. Louis University, USA.
So this is the first time for me to come to Chiang Mai. Actually, I have several times to Bangkok, maybe Hong Kong before. So today, maybe my topic's nothing about the autism, but I just want to tell something about the gene test and its applications in for the human life. Okay. So my topics about the gene test and its use for the children development, maybe for the human health. And my name is Ming Xing. Ming Xing Ma. Ma is my family name, and I'm the CEO of Huanchi Group. Yes, so my story will start from the precision medicine. Yeah, I think this word is quite familiar with all you guys because it's just announced years ago by the president of the USA. I think maybe the precision medicine just talk about two things, and how to diagnostic and how to make the treatment. So for the diagnostics, we need to get some method to get the key or maybe the real reason of the disease. And then based on the diagnostic, we can take the treatment. Fortunately, I think maybe recent years, maybe in the decades of years, the treatment has been developed very quick in the hospitals. But for the diagnostic, maybe we just use some very old, very traditional ways to take the diagnostics. So I think maybe the bottleneck of the current healthy solutions will be the diagnostic. And also I think the test take the core role of the several things like the disease prevention or the therapy and also for the bioresearches. So we just start our project based on the test. Okay. So the current situation of the clinical diagnostic is just as listed. So for the technology, we just use the biochemistries or immunologists and maybe cultures for some microbiomes. And it has long years of history, and it's very simple to use. Maybe it's very easy to use and also well used in all of the hospitals. But the performance of the current diagnostics is not very good. Like for some of the specificity and sensitivity is not good. Like so maybe when we test like the HPV or HBV infection, we can only test maybe 60 or 70% of the sensitivity rate. And also the output is very low, very low. So when we go to the hospital, during one single test, we can only get one result and maybe correlated with the disease or not. And so we must try to several times to do the test and to get tons of the report for one disease. So this is the current situation. So the molecular, so because my major is molecular biochemistry and molecular biology, so I just want to make the researchers to be application. So I think the nucleic acid is a very good target to be taken for the diagnostics, because every the biology, each of the the pathogen or human or the animal has the nucleic acid. So it's very to extract, and also it's very easy to be tested. And based on the PCR or other kind of the application methods, we can do the amplify to tons of the folders, so we can see very, very tiny and also very, very earlier. So also we can use the several test to get a higher test threshold. So that means we can test like very early of the disease. But the current situation is in the market, there's no more kind of the technologies just based on the high throughput DNA test. So that's why we just can do the PCR or the qPCR in the hospitals, but we cannot get any of the high throughput DNA technologies. So if we can have something high throughput, we can get more information from the test and give more information to the hospitals, to the doctors, also to the patients, and also make more things to the researchers and development. Our project was based on the microarray technology. So the microarray is not something new, it's quite old. It's just maybe launched around the year of 2000 based on the Human Genomic Project. So maybe during that time, there's no NGS till that time. So they need to think about the method to treat the tons of the data in a short time. So the microarray just born. And the microarray actually, here's some very simple principle of the microarray. So for the microarray, we just spot some of the target probes, we call it probes, on the slide. So if we want to test the blood, if we have the HAV, HBV, HCV, or HDV, we just clone some of the probes and then spot on the slide to form as a microarray. And then each of the probe is a fragment of the specific DNA of the pathogen or something. Then, when we want to do the test, we just collect the samples from the patient and then do the PCR, and along with the labeling by maybe the 5' of the primers, and then make a hybridization between the PCR product and our microarray. After that, we can wash out the non-binding probes and then to scan by the laser or by other CT systems. Then we can see some of the spots. These spots will infer to the infection or the SNPs. Also, the brightness of the spot will refer to the infection level or the quantity of the gene. This is quite simple. So during this whole processor, the specificity and sensitivity are all ensured by the PCR and all by the hybridization. And I think also we can add more controls into the whole test. Because in the experiment, I think maybe control is quite important. When someone to do the experiment, like we get some samples from the blood from the patient, we must sure the sample must be the sample from the human. Maybe there's a story in China. Some guy just take some tea to take the test of the urine, so they get the same report. I think it's possible because the machine cannot recognize the two samples. So for us, we can add more like the inner controls to ensure the whole test of the PCR or the sampling, and also we want to keep the quality of the chip too. So maybe this will increase the reliability of this whole system. Okay, this graph just shows the whole procedure when we got a sample from the first stage and to the report at around 5 hours. And this can just finish in our professional lab, and also it can provide a whole report about the whole test. It's quite soon, I think, compared to the current technologies. Okay, so for the use of the technology, what we can gain. So maybe this one is most important, the screen test. So when we go to the hospital, if I got a cough, and I ask the doctor, what I got, maybe what infect me. So the doctor will guess which will be the most possibility, and then to take the test to see if I'm correct or not. If it's not correct, so take the test again. So the screen model means no matter what I got infected, just take my specimen and then to take the test and see the report to get the final answer, and then treat me based on the report. So this is quite important for the current doctors, because in China, the level of the doctor is quite different from maybe big cities and maybe some towns or the countrysides. So if we want to make every doctor at the same level, it's not possible. So we can just improve our test to make them equal. So maybe we can benefit more from these two. Okay, so what can the gene test do? I just can say three aspects. The first one to test the presence of the genes, that is the infections. If I can infect it by something, the gene will in my body, so I can test them, and then I can see you have got the presence of the pathogen gene. And secondly, we can test the genotypes of the gene. Like now the HPV vaccine is very popular in these recent years. So HPV typing is also very popular. So we have designed a test to test 40 types of the HPV. So this can just help the doctor to see, what kind of the genotype of the HPV have you got. And also, the genotyping is very accurate, more than other typing methods, like to screen by observe by something. So the second use is the genotyping. And third is to test the mutations in the genes. So I think maybe mutation will occur just frequently in our body, and mostly of them are maybe non-harmful. But some of them will be related to the genetic disease, and some of them will be correlated with the PGX. That means this will affect some use of the drugs or our lifestyle. So I think maybe the gene test can be used these three aspects. And mostly maybe recent years, we can use to count the genes by the NGS, like for the NIPT. It's also a new way to use. So Huanji is a very small company. I built it maybe 10 years ago. And it's based on the gene chip technology. So we built a platform in China, in Kunming. We have a workshop there and get a certification of the CFDA, of the GMP certification. And then we use the printers, print the chips, and then we have the whole reagent solution, and also for the devices involved in this test. And for the years of the development, we have formed 6 lines of the productions. First is the systematic infection disease detection. This may be caused by the digestive, maybe the urodactyl, and maybe somewhere maybe blood, fungi. So this will help the doctors to solve the sets of questions caused by the infections. And then we have the personalized medicine production line. This will guide the people to how to use the drugs, maybe the folic acid, maybe some hypertension, or the diabetes, etc. And third one is for the pathogen drug resistant, missing one word. So something like the TB or HPV and CV has the resistant problem for doctors. So we can just detect the fragments just mutate against the drugs, so we can test them in a very short time, especially for the TB. And also we have the genetic disease test for the Y chromosome micromycin for the males, and also we have something for the hereditary deafness or the other things. We call it single gene mutation hereditary disease. Okay, and then also we have the tumor PGX. In this section, we have 2 kinds of the test for the chemotherapy and targeted therapy, 2 ways. So for the chemo, just to avoid any of the risk to use the chemo drugs to maybe improve the efficiency of that. And also for the targeted drugs, just to evaluate which kind of drug will be quite useful, maybe non-function to the body. So this will reduce the cost of the patient, also will increase the treatment too. And finally, we have a prediction section. This section, we just want to make some entertainment gene test section. So this is just for some assurance, maybe predictions for your lifestyle, maybe your talent, maybe something your fatness, or obesity, or maybe something your skin's characteristic of your skins, something. So this kind of technology is just used for the evaluation, maybe just for our lifestyle, not for the clinical use. Actually, in my mind, the gene test is not something, but maybe in the hospital, we cannot use that very rough, maybe very precision, because in the hospitals they do not have very professional labs and maybe do not have very personal teams, because one of the characteristics of the gene test is sensitive. The sensitive is good for the test, it's also harmful for the operation. So when we just do the PCR, if even a single PCR, the false positive will occur frequently, maybe in China, tons of the years. So the government has set many rules to limit it, such of the development of such laboratories. So based on this, the demand of this, we have built maybe now it's eight laboratories around China to provide the diagnostic services to the hospitals or to the individuals. Because we have the whole solution of the PCR and maybe higher throughput technology, so we will have a better organization of the whole thing, so we can provide more accurate test. And now in Thailand, we have another new, from 2015. We got introduced by the Thai BOI and also by the TCL of the Thailand. So we have a lot of discussion with the government to make some really solution for the gene test. So very appreciate for Thailand has such an opportunity for our company, for our Polisa to be here to do this. And okay, so maybe here give some example for my the solutions. This is for the infected test, like for HPV typing, for some disease detections, like for the fungi. And maybe today the topic is mostly for the children, and I think maybe in the red box is mostly for the children too. So for these two kinds of the infection disease are all maybe commonly occurred for the child, maybe for the old people's like the ICU or some special department. So we can test like 30 pathogens, include the virus, the fungi, and the bacterial, and maybe RNA and the DNA together at one time, around 30. So finally, after the test, we just get a sputum, and after 5 to 6 hours, we will get this report to see what present in your sample. And then the doctor just see the report and to get the medicine is okay. So for this, we have maybe about 300 to 400 samples per day for each of our clinical lab. So we had contact with the hospitals. They said after they use the stage of the service, they can have a very quick routing of the beds in the hospital. So they can treat more patients in a very short time because the test is very accurate and also rapid. So this is for the infections. And also for the personalized medicine, it's also very important, maybe also interesting. Because the personalized medicine, actually the principle is quite simple, because these enzymes are based on maybe the target or in charge of the transportation of the drugs, maybe or take response to the metabolization of the drug too. So we have a special test only for the maybe child, for the children, for the safety use of the drugs. As some of the kind of child in China, when they was very young, very little, they use some kind of the antibiotics. They got a deafness. So in China, this is quite a big problem. So China government just launched several projects to make the screen of the deafness genes for the little child. And also for this, we want to do this too. With this test, we can just screen the deafness genes, maybe present in the little child, and also guide them how to use other drugs. Got the fever, got the pain, and how to use, and the dose, and some side effect, so etc. I think maybe this is a new way to help the child too, help the children too. Okay, what's that? Okay, so for predictions. Maybe mention, as we mentioned for gene test, maybe as we mentioned for the NGS, we always think about the cancer, the tumor. Because the human want to detect the tumor or the cancers earlier, it's very urgent. But actually, maybe unfortunately, we don't have no more ways to test earlier. We only have the pathology method to maybe ensure a tumor occurred. So we call this kind of test maybe the prediction, or maybe it's not very serious, it's in an entertainment level. So we can test like the skin or the optics, or maybe even the children talent based on the sum of the genes. For the principle of the test, I think maybe there are two ways. Because some of the gene is, you can see, take it as the tag. Maybe it's a marker for some of the group of the specificities. Like, okay, we have 10, maybe 100 of the children. 10 of them has some talent in the musician, but the others do not have. So we will analyze the gene typing of them to make a wild, maybe a whole, like the sequencing of them to see, oh, maybe he has a marker there. So we will take it as a musician gene. So for some of the other things like the actin 3, it will be take two like responsive to the muscles. The first type should be very strong and can last long, and maybe for the second one should be very strong but can last short. So with this test, we can tell them maybe you are good for a long run, for the marathon, but the otherwise for the short runs should be better. So this can be also taken for the kindergartens and for some guidance for the parents too. But I think this very easy test and it's not very serious, so just use it as the entertainment too. So for our the services, we have served for many ways for the hospitals, institute, and universities to do the test. Also, we can just develop some new things. Like we can have some cooperation with other guys to make some new kits, and also we can develop some individual market. This can be some individuals just have some problem with something, but maybe it's very hard to talk to others. We can just give the sampling kit to them, and then after a few days of the waiting, they can get a personalized solution. So this is our two lines of the production. And so finally, we can get very many benefits from our test. Maybe here, lower the boundary and also get a cost efficiency too. So for the test of the gene, we cannot neglect the NGS. The NGS is maybe is more famous than the gene chip or the microarray. I think maybe for the NGS, it's more suited for do the research or to do something like the cancer, to do the CFDA detection for some of the cancer mutations. It's very good to use that. But for some use of the common detections like the infections or the PGX, I think the microarray is more suitable because the cost of the microarray is very low. We can just do it by several hundred yuan, or maybe in China. Okay, it's very easy to be accepted by the common guys, and also we can get the result in few days, maybe 1 or 2 days. So in China, each of the lab, we just can output the report during 1 workday. That's quite important for the doctor to save the patients. And furthermore, I think maybe we can have an open platform based on the microarray. We can have more chance and more ideas to develop something new with the method. Just with no more high throughput, we can just test several hundreds level of the test at one time. So we can have more chance to extend this platform too. And also, I think the microarray is a very useful tool, but it's only a tool. This maybe we cannot tell more story about with the gene test, but we can definitely to solve some very basic demand of the current situation of the hospital or by the patient. So we can not only tell the story, and also we can do something did, okay? So maybe my talk is very short, and maybe because it's my first time to speak in such an international international conference, so maybe I have no more experience, but I think I want to share my mind to you guys here, and also we want to provide more services and have more like the cooperation in the future. So thank you so much. Okay.
Thank you for your talk. We realize that China is very rapidly growing in the field of genetic microarray and also whole genome sequencing. Today we are talking about autism. Who is your partner in China that collaborate for you to develop? I saw that you have few genes with respect to children, for intelligence, for things like that. But as I found out at preparing my lecture, and I discussed this morning, with regard to autism, we have not less than 300 markers that are found to be different from normal phenotype. I'd like to invite you to comment that would there be in the future one microarray gene chip that you would develop so that we could screen the whole entire genomes for things like autisms, like Fragile X, and all, you know. Fragile X is the most specific because we know or Rett syndrome, you know, which we already have, and also we need a quick solution. At the moment, we use the quick solution is very expensive. And very short path. We use fluorescent antibody, specific fluorescent antibody for the epitope sequence, and with one drop of them on the fresh blood, in 5 minutes we could read whether we have the signal, and it gives us the positive or negative. For example, we could screen Fragile X with Fragile X mental retard protein FMRIP1 in 5 minutes, just one drop. But the antibody is damn expensive.
Yeah.
Okay, because it has to be developed in a very, very expensive way. My question is that China is one very fortunate because the number of people you have in China which could benefit from this, and therefore you have much larger platform, and the price could be divided thing. Okay, I'm very glad to see that you are going to develop with T-cell. We are also looking forward to T-cell to invest or co-funding in many research projects. With regard to Fragile X, I'm not sure if you know that you can get a lot of good material from so-called Simon Simplex. You know the word Simon Simplex? Simon Simplex is supported by Simon Foundation, a large Jewish-based supported foundation in New York. The head of them is Gerald Fishback. Gerald had been working to find out what caused autism for many years, and he gave many lectures in Thailand. Simon Simplex is the material from cohort of family which have genetic or dominant inherit genomes, you know, so that you can really find markers that are inherit. Okay. Yeah. So two questions: who are your partner in China with regard to autism, and how you could really develop these autism chips? Thank you.
Okay, so I have no partner there, So maybe for our test, I just take it as a tool. Maybe it's a converter from research to application. So we do not make any of the deep scientific researches maybe based on the many disease. We just use the very these informations to develop the tools. So maybe in the future, based on some new findings of this field, we can just convert them to the new tool. Why I was here to announce this? Because I think maybe in hospitals, this just do the things with the technology very old, maybe 6 years, maybe 10 tons of years, maybe 1 decade. But in the research level, I'm also do the research when I was in the university. I think maybe it's higher, maybe far more higher than the current use. So I should find something middle, just can connect to the doctors and also connect to the use of such of the technologies. So that's why I do this. I did this for a long time. As you mentioned, maybe some of these are quite complex, right? It's not one single gene. Maybe it's a gene and the environment co-infections, co-reaction. So this will be a very, very long term to make the effort to do that. So if we can get something new and maybe some markers has been developed or announced, so we can just easily transform them into the application. That's my aim of the project. Thank you so much.
Okay.
Thank you.
Thank you very much, Dr. Ma. And we will later on discuss how we can collaborate from Thailand and China later. Well, before we have a coffee break, I would like to call upon my colleagues from Greece, Christina and Nadia. And the reason is simple. Before lunch, I think Christina gave me the medal, okay? And I'd like you to tell the audience why you give it to me and how could you support our autistic project in the future. Maybe please come forward, both of you. Thank you.